Hi there. In this video we are going to discuss about the secondary structure of proteins. In previous video we discussed about the primary structure and how to determine the primary structure. So primary structure was the basic sequence of amino acid in a protein or polypeptide chain, while secondary structure of protein is the next arrangement of that polypeptide chain. So that polypeptide chain does not exist simply. Instead it attains different structural conformations, different 3D structural conformations which assume its secondary structure. And these different structural conformation of secondary structure include alpha helix, beta pleated sheets, and beta bands. So these are three principal secondary structure of proteins, and among these three principal secondary structure of proteins, they may arrange themselves and form super secondary structure, which are motifs. So these are the different secondary structure of proteins. So what is alpha helix? Alpha helix, basically this was assumed this is a simple polypeptide chain composed of amino acid sequence or amino acid chain. So it assumes a spiral formation. And this spiral formation of this polypeptide chain is called alpha helix. This is alpha helix of the polypeptide chain. And this alpha helix, on this one turn of peptide chain, there are 3.6 amino acids. And it is stabilized by hydrogen bonding. There is hydrogen bonding between amino acid chains here, and this hydrogen bonding occurs principally. We know that amino acid has carboxylic group COOH as well as NH2 group, and this carboxylic and amino group impart the polarity to these chains and polarity to the amino acid. So they have polarity, they have oxygen, and they have hydrogen. This oxygen is partially negative and hydrogen is partially positive usually here. So this partial positive and partial negative charge present in the amino acid chain lead to the formation of hydrogen bonding among the amino acid, and this hydrogen bonding stabilizes this alpha helical formation of the proteins, which is its principal secondary structure. So which amino acid disrupt this regular structure? This regular structure is disrupted by amino acid. For example, proline. We previously discussed in the structure of amino acid that proline is an amino acid. Which is having a ring structure in which nitrogen is having two bonds with the carbon atoms. So proline is an amino acid, and due to its amino nature, it does not have a proper amino group like NH2. So due to this reason, proline disrupts the regular structure of the alpha helix and induces a nick here, or induces a bends in the structure. Moreover, the charged amino acid also interfere with this structure due to the charged nature. For example, glutamic acid, as well as the aspartic acid, as well as the basic amino acid, for example histidine, lysine, they have additional charge or charged groups. Their charged groups interfere with the regular bonding of alpha helix and disrupt the structure of alpha helix. So this alpha helix is found in both fibrous protein, for example keratin. It is present in fibrous protein, for example keratin. It is also present in the structure of globular proteins, for example myoglobin. So it is present in both in fibrous as well as globular proteins. So it is about the alpha helix, and it is a 3D structure. Stabilized by hydrogen bonding. So next we move on towards the beta plated sheet. Second secondary structure is beta plated sheet, and beta plated sheet is the sheet formation. It is a structure made up of sheet like a paper sheet. If you bend it again and again, and you attain this configuration of the paper here. So this is a sheeted configuration of a paper, like a paper, and it is formed usually of either of a single peptide or of multiple peptides. You can have single peptide or multiple peptide. How single peptide form beta plated sheet? A peptide chain can fold upon itself and form two arms, and these two arms then interact by hydrogen bonding and so on, and these interaction lead to the formation of beta plated sheet. So this beta plated sheet, it can be parallel as well as anti-parallel. it can be parallel or anti-parallel. Supposedly these are two polypeptide chains which are forming this sheet, and this sheet is being stabilized by hydrogen bonding here. So if both these are parallel, N terminal, C terminal, N terminal, C terminal. So if you both N terminal on same side and C terminal on the same side, then it is parallel beta platelet sheet. This parallel, if they are opposite, supposedly here is N and here is C, then it is anti-parallel beta pleated sheet. So beta pleated sheet can be parallel, it can be anti-parallel, and it is stabilized by hydrogen bonds. It can be formed between single polypeptide chain, or it can be formed between the multiple polypeptide chains. So in illustration, we show this beta pleated sheet by an arrow here, by an arrow. For example, if they are parallel, they are shown by arrow in same direction, and this arrow is joined by a stretch of protein and a stretch of amino acid. And if they are anti-parallel, then they are in opposite direction. They are anti-parallel, then they are in opposite directions. So in this way we indicate this beta pleated sheet. Anti-parallel in this way, opposite direction arrow, and parallel arrows are in the same direction. Parallel arrows are in the same direction. So it was about the beta pleated sheet. Next our topic is beta bands. Next our topic is secondary structure is beta bands, and beta bands basically induce a sharp turn in polypeptide chain. For example, this is a polypeptide chain going on, and here it want to turn about, then beta bands induce the turn here. Okay, so beta bands are responsible for inducing turn in the structure of polypeptide chain, and these beta bands also induce a form a secondary structure, and these beta bands are usually rich in the amino acid, for example glycine and proline. Glycine and proline, previously discussed, proline interferes with the regular structure of protein due to its amino group. So glycine and proline are found in the beta band, and these are usually found in the surface of protein and induce the bands on the protein structure and induce the regular folding of the protein chains. So these are the beta bands. Next we move on towards the non-repetitive secondary structure. These first three structures are highly repetitive. They occur again and again in the protein structure. However, there are certain structures which are not repetitive so much, and these are called non-repetitive structures. For example, this is the beta pleated sheet, and this is alpha helix, and these are joined together, and so on. So they form a structure. Then between here is another alpha helix, then beta pleated sheet, then again alpha helix, and so on, and here is another structure, and between this structure there is some loop, or here can be a coil. It can be a coiled structure or coil. So this coil or loop which is less repetitive than this beta pleated sheet or alpha helix is called non-repetitive secondary structure, and they usually exist in the form of coil or loop. Next we move on towards super secondary structures or motifs. These motifs are called super secondary structures. And what does they mean? Simply, if we define motifs, these are basically combination of secondary structures. These are basically combination of secondary structures, and they are between secondary and tertiary. They are between secondary and tertiary configuration of proteins. So motifs or super secondary structure are between secondary and tertiary structure, and they are formed by a combination of different secondary structures. For example, this is a beta pleated, this is the beta chain, this is also beta strand and beta strand, and these two beta strands joined by alpha helix. These are beta alpha, this is a beta alpha and beta. So it is a combination of secondary structures. This is a motif. Similarly, here is a alpha helix, this is alpha helix, and they are joined by loop here, and they arrange a regular configuration here, and they can be regarded as a combination of two alpha helix, so they can be regarded as motif. Okay. Similarly, beta D is also a motif, which is a combination of multiple beta pleated sheets. So multiple beta pleated strands join here and form beta D, which is also a super secondary structure motif. Similarly, beta barrel, which is combination of many beta pleated strands, it is called beta barrel, which is also a motif. So these motifs are found in the structure of proteins and introduce the complex nature of proteins. So it was about the secondary structure of protein. Next we move on towards the tertiary structure of protein. Next we move on towards the tertiary structure of protein. So what is tertiary structure?